Biofilms. 1. Introduction. Biofilms are communities of microorganisms, e.g. bacteria, protozoa, algae, fungi, which attach to surfaces and are embedded in a matrix of highly hydrated extracellular polymeric substances EPS. Globally, more than 90% of all microorganisms are organized in biofilms and are found in ecosystems such as lakes, surface water, groundwater, and terrestrial ecosystems. Biofilms can also settle in ecological niches under extreme living conditions concerning hydrodynamic and osmotic stress and pressure, temperature, as well as exposure to extreme pH values and chemicals, e.g. biocides and antibiotics. Biofilms are key players in numerous microscale ecosystems, e.g. subaerial biofilms or marine biofilms. Biofilms can also persist at unwanted locations, e.g. clinical instruments, causing tremendous problems as they can host pathogens triggering infections and diseases. In technical systems, the presence of biofilms can be critical as well. One example is biofouling of membranes in water purification processes. On the other hand, biofilms enable technical applications in e.g. biological wastewater treatment, biotechnological productions of chemicals or drugs, and bioremediation of soils and groundwater. Biofilms are subject of interdisciplinary research, merging biology, chemistry, analytics and engineering. Even though biofilms are ubiquitous in natural and engineered systems, they still need to be further studied and understood. Fundamental mechanisms need to be investigated in detail to learn more about their applicability and benefits in environmental and technical processes, but also from a holistic point of view regarding material flows and overall turnover, in the light of green chemistry, too. One valuable analytical approach is nuclear magnetic resonance, NMR, among others shortly summarized in the following. As most biofilms are multi-species cultures and, therefore, are highly heterogeneous with respect to structure as well as biological and chemical composition, there hardly exist standardized qualitative or quantitative analytical methods for detailed and comprehensive measurement and characterization of biofilms. Imaging techniques significantly contributed to a better understanding of structure, functionality and their interplay, as summarized in numerous international publications, one example being 19. Optical microscopic techniques as light microscopy, confocal laser scanning microscopy (CLSM), or Raman microscopy, but also electron microscopy revealed physical structures and morphologies as well as chemical compositions of biofilms on different length scales. Electron microscopy can image biofilms at high spatial resolution, but has limitations due to its invasiveness and destructive sample preparation, e.g. drying and application of vacuum. Geometric parameters as biofilm thickness and coverage can be obtained, however resolution, contrast and invasiveness of the named imaging techniques represent the major limitations. CLSM techniques offer the possibility to investigate the biofilm structure and its components on the sub-micrometer scale of spatial resolution. However, specific staining agents are necessary, and only a selective image can be obtained. New analytical approaches, for example, optical coherence tomography, October, non-invasively revealed the development of biofilms and their mechanical properties, for example deformation, with a spatial resolution down to a few micrometer. Biofilms in medicine are complex communities of microorganisms that adhere to surfaces and are embedded in a protective matrix. They play a significant role in various medical fields, including infectious diseases. 1. Chronic wounds. Biofilms delay healing, resist antibiotics. 2. Respiratory infections. Biofilms in cystic fibrosis, pneumonia. 3. Urinary tract infections, UTIs, biofilms on catheters, bladder walls. 4. Dental plaque. Biofilms cause tooth decay, gum disease. Implant-related infections. 1. Prosthetic joints. Biofilms on artificial joints cause infection. 2. Cardiovascular devices. Biofilms on pacemakers, heart valves. 3. Contact lenses. Biofilms cause eye infections. Chronic diseases. 1. Diabetes. Biofilms in foot ulcers, wound infections. 2. Cancer. Biofilms promote tumor growth, metastasis. 3. Alzheimer's disease. Biofilms in brain tissue. Biofilm formation. 1. Adhesion. Microorganisms attach to surfaces. 2. Microcolony formation. Microorganisms multiply. 3. Matrix production. Biofilm matrix forms. 4. Maturation. Biofilm becomes structured, resistant. Biofilm characteristics. 1. Antibiotic resistance. 2. Evades host immune system. 3. Persistent infection. 4. Difficult to eradicate. Treatment strategies. 1. Antibiotic combinations. 2. Enzymatic disruption. 3. Photodynamic therapy. 4. Nanoparticle-based treatments. 5. Surgical removal. Research areas. 1. Biofilm prevention. 2. Biofilm disruption. 3. Antibiofilm coatings. 4. Personalized medicine approaches. Key researchers. 1. Dr. Philip Stewart, University of Montana. 2. Dr. Garth Ehrlich, Drexel University. 3. Dr. J. William Costerton, University of Singapore. Journals. 1. Biofilm. 2. Journal of Bacteriology. 3. Infection and Immunity. 4. Microbiology. Conferences. 1. International Conference on Biofilms. 2. Biofilm Research and Applications Conference. 3. American Society for Microbiology Conference. Funding agencies. 1. National Institutes of Health, NIH. 2. National Science Foundation, NSF. 3. European Union's Horizon 2020. Staining techniques are being developed to differentiate bacterial cells growing in living animals, e.g. from tissues with allergy inflammations. Research has shown that subtherapeutic levels of beta-lactam antibiotics induce biofilm formation in Staphylococcus aureus. This subtherapeutic level of antibiotic may result from the use of antibiotics as growth promoters in agriculture, or during the normal course of antibiotic therapy. The biofilm formation induced by low-level methicillin was inhibited by DNase, suggesting that the subtherapeutic levels of antibiotic also induce extracellular DNA release. Moreover, from an evolutionary point of view, the creation of the tragedy of the commons in pathogenic microbes may provide advanced therapeutic ways for chronic infections caused by biofilms via genetically engineered invasive cheaters who can invade wild types, cooperators, of pathogenic bacteria until cooperator populations go to extinction or overall population, cooperators and cheaters, go to extinction. Pseudomonas aeruginosa. P. aeruginosa represents a commonly used biofilm model organism since it is involved in different types of biofilm-associated chronic infections. Examples of such infections include chronic wounds, chronic otitis media, chronic prostatitis and chronic lung infections in cystic fibrosis (CF) patients. About 80% of CF patients have chronic lung infection, caused mainly by P. aeruginosa growing in a non-surface attached biofilm surrounded by PMN. The infection remains present despite aggressive antibiotic therapy and is a common cause of death in CF patients due to constant inflammatory damage to the lungs. In patients with CF, one therapy for treating early biofilm development is to employ DNase to structurally weaken the biofilm. Biofilm formation of P. aeruginosa, along with other bacteria, is found in 90% of chronic wound infections, which leads to poor healing and high cost of treatment estimated at more than $25 billion US every year in the United States. In order to minimize the P. aeruginosa infection, host epithelial cells secrete antimicrobial peptides, such as lactoferrin, to prevent the formation of the biofilms. Streptococcus pneumoniae. Streptococcus pneumoniae is the main cause of community-acquired pneumonia and meningitis in children and the elderly, and of sepsis in HIV-infected persons. When S. pneumoniae grows in biofilms, genes are specifically expressed that respond to oxidative stress and induce competence. 114 Formation of a biofilm depends on competence-stimulating peptide, CSP. CSP also functions as a quorum-sensing peptide. It not only induces biofilm formation, but also increases virulence in pneumonia and meningitis.